Hello and welcome. This is Bob Lessig at Johns Hopkins. In this lecture, we will examine primary and secondary keys and how they operate as unique identifiers. We will look into relational databases. By connecting smaller databases, information can be divided without an excessive burden on a single database. Most biological databases are set up in this manner. I recognize that many of us are coming from very different backgrounds. For those very familiar with databases, this should serve as a pretty short review. For those less familiar, I hope to give you a good introduction. After viewing this lecture, we should be able to define some terms, such as field, record, relational database, as well as primary and secondary key. Be able to find a primary key when you look at a database. It's usually an identifier. It's usually a series of numbers or letters. It's not data. Look at the setup of a relational database, which is a series of linked databases. Finally, explain how a secondary key in one database can serve as the primary key in another database, usually a linked database. One of the simplest forms of a database is a spreadsheet. It's basically a grid with rows and columns. There was an old spreadsheet program called Lotus 1-2-3 for those who remember. The most commonly used spreadsheet today is Microsoft Excel. Usually, the columns represent a particular type of data. That is a field, one of our key terms for this lecture. The column header usually describes the field: name, birth date, account balance, a particular statistic. Fields are usually specific for certain types of data. For instance, a date field might be set up in a desired format, like month/day/year. We can then call the rows records, sometimes known as entries. Essentially, the terms are synonymous for this purpose. A record might be an individual person in a directory, or it might be a DNA sequence in a biological database. Our last definition is of a primary key. Each record in a database should contain a unique identifier. It's often numerical or alphanumerical. Alphanumerical simply means mixed letters and numbers. There should be no duplication when it comes to a primary key. One identifier should represent one and only one record. We will see examples shortly. Here is some data from United States presidential elections. There are 7 columns. Cand is an identifier for a presidential candidate. Cand/year is an identifier for a candidate in a particular election. Candidate is the name of a candidate. That's data. That's not an identifier. Year is also data. Party represents data. For which party was that candidate affiliated? EV stands for electoral votes. The candidate with 270 or more is the winner of that election. Pop vote stands for popular votes, which does not decide elections. The first question to think about is, what is the primary key in this database? You should only have 2 choices. Only 2 columns are identifiers. Those 2 columns are marked Cand and Cand year. How are they different? Which is the primary key? The primary key is actually Cand year. For instance, 1992 D1 means the Democratic candidate in 1992, so that's William Jefferson Clinton. Each record represents a vote total in a particular year, so Cand year is unambiguous. Cand is what we call a secondary key that represents one candidate. That candidate could have run more than once. So we can see how it differs from Cand in uniqueness. The Cand identifier represents the candidate, and Cand year represents a candidate's vote total in a year. So it's more specific to the record in this case, and is therefore a primary key in this database. Here is one of the most popular databases on the internet, and I'll use it to illustrate in a crude way how to set up a database. That's the Internet Movie Database. Many of you are probably familiar with IMDb, but if not, it's a very commonly used website, and it's basically a relational database. This is the movie page for the 2014 movie Godzilla. Now, there are many movies with the title Godzilla, so how do I click the right one? Well, here's where you could use a primary key. And when you click the link marked Godzilla 2014, you are actually accessing a primary key without even knowing it. The primary key is right here on this page if you know where to find it. Where is that primary key? It's right in the URL. You see imdb.com title/tt, and then it's small, but you can find it yourself on the web. You see the identifier 0831387, and essentially that's a primary key in the movie title database. Now, a movie database might look something like this, but I've really oversimplified it. I'm not going to suggest that IMDb is based on a simple spreadsheet, but let's take a look at this data on the bottom. The primary key in this case is ident, which lists an actor's work in a particular movie. A secondary key is movie ID. So you see movie ID, that's the number we saw on the last slide, 831387. I left off the preceding 0. That's a primary key in the movie title database, but if this is a movie role database, that would be a secondary key because there are several actors in the same movie. I only listed 3. You might also notice that actor ID is also a secondary key. If you notice, the 300 identifier appears twice for Juliette Binoche. The movie ID fields, essentially in IMDb, become accessible through links, so that when you click on Godzilla 2014, you're really clicking on the URL that has the 831387. You're actually clicking on a primary key in the movie title database. You would be clicking on a secondary key in this particular database. So let's look at that database that we've already seen. That's at the top. And in particular, let's look at the actor Aaron Taylor Johnson. Now, we don't want to put all of his information into this top database because this is movie roles. That might become a little too cumbersome. So we take that secondary key in the top database and make it a primary key in the actor database at the bottom. So we see Aaron Taylor Johnson was born on June 30th, 1990 in England, and you could get a whole bunch of other information into that database, but that's the basic idea. We can link databases together by taking a secondary key in one database or in one relation and making it a primary key in a related database or relation. So basically, IMDb, like the biology databases, tend to be relational databases, and the idea is to avoid too many fields in one database. The goal is to link smaller databases together. Each smaller database can be called a relation. The concept is that a secondary key in one database can serve as a primary key in another database. So if you're on those movie roles and you click the actor ID, that might link to the actor database as a primary key. We can think of the internet as one large relational database, because every time we access a page on the internet, essentially it's a text file, sometimes with a lot of markup. Whenever you click on a link, you're clicking on a primary key to the other database, which is that website which contains its own set of information. So URLs essentially represent primary keys. Think about biological databases. On a DNA sequence record, we might not want to put every single thing known about this DNA sequence into one single record. So we could link to a protein database to access proteins coded for by this DNA sequence. Or maybe a structure database for that protein structure. Or a scientific literature database for papers written about this particular piece of DNA. Relational, or connected, databases come in very handy. NCBI is a frequently used website and database in bioinformatics. It is one of the most used websites in the world with millions of hits daily. It is based at the National Library of Medicine at the National Institutes of Health in Bethesda, Maryland. What's there? Well, go to the website and take a look around. You will find databases for scientific literature, DNA sequences, organism taxonomy, gene expression data. Well, quite a bit. It's a huge relational database. So basically, to summarize, be able to recognize the primary key. That's the most unique identifier, and it's usually a series of numbers and letters. It's not data, so don't look for instance, a vote total and say that's a primary key. And relational databases are able to link related data. In the case of IMDb, it's a movie database that can be linked to an actor database. But at NCBI, we can see that a nucleotide database can be linked to a protein database, and also linked to a literature database, and a structure database, and a host of other databases. I hope this lecture on databases is helpful to understanding the concept of databases, and good luck.
Hello and welcome. We'll be looking at searching PubMed effectively. PubMed is a government website based at the National Library of Medicine. It contains over 23 million biomedical and life science publications. Searching it effectively can be a very useful strategy. It is also relevant because the mechanism for searching PubMed is remarkably similar to the method for searching nucleotide and protein sequence databases.
Our objectives include searching by author. For instance, finding papers written by a particular individual, either by using the last name format or by using one or two initials. We will also look at how to search with field tags. In other words, limiting your search to very specific fields in PubMed. We will search by subject and combine searches with subject and an author name. That is a Boolean operation. We will look at the MeSH database. MeSH has a controlled vocabulary that allows for more effective searches of PubMed. We will use filters. Filters help narrow down to certain types of papers. We will also examine the truncated searches, which allow for searching for variations of a particular word. First, let's go to PubMed. There is an address for PubMed at the National Library of Medicine. However, PubMed.gov, that is PUBMED.gov, is a working alias, so PubMed.gov will take you to the NIH website where PubMed is hosted. Let's use the word GRIDER and search. You will see some papers that include an author named GRIDER, which I just highlighted. You will also see some papers where GRIDER is not listed as an author. Carol Greider is a Nobel Prize-winning researcher of telomeres. So perhaps Greider was mentioned in that particular paper. We'll look at a way to avoid those kinds of results shortly. The last name Greider finds 223 results in PubMed. If I want to limit further to specific authors with the last name Greider, I can add a first initial. So Greider C would narrow down to 102 papers. You can see that Carol Greider's second initial is W. A one-initial search will find all variations of C, so C followed by any second initial, or just C by itself. I could further narrow by adding the second initial. You see that narrows the search down to 94 papers.
So basically, last name is the broadest search. One initial is a little more specific, and two initials are the most specific. Some authors use more than two initials. However, PubMed limits searches to two initials, and PubMed actually only enters two initials for any author.
Okay, now I mentioned there is a way to avoid matching words outside the author field. I'm going to go back to searching for Greider.
last name only. If you noticed, the first paper did not have Grider as an author, so I'm going to open that paper now. When you open a record, it's going to show the abstract. So here's the abstract, and you can see that it says that the 2009 Nobel Prize in Medicine was awarded to, there it is, Blackburn, Stozak, and Grider. So her name is in the abstract and not in the author field. The author field lists only one author, and that is Wang. So, if I want to limit the search to only the author field, the way to do that would be to type Grider, bracket, author. Now, I only see those papers where Grider is in the author field. I eliminated that result and others where Grider was in the abstract. Basically, by putting brackets around the field name, I can limit a specific term to a particular field. There are acceptable abbreviations. For instance, if I had typed bracket AU, AU is an accepted abbreviation for author. I would get the same result. There are a list of fields available that are searchable, including the author field. Now, suppose I wanted to search by subject. There are several ways to do it, and usually one of the easiest ways to do it is to simply type in the subject. So I'm going to type in RNAi. RNA interference is a very, very popular search topic and a very important topic in life science. RNAi was famously studied by Craig Mello and Andrew Fire. So let's say that I want to get papers on RNAi by Craig Mello. I can type in RNAi and Mello bracket author. Now what you might notice is that and is capitalized, and for all searches you should capitalize the Boolean operators. Boolean operators are and, not, and or. This gets me papers that are about RNAi and are written by an author named Mello, which gives me 27 papers. Some of them have RNAi in the title. Some of them don't. So, if I want to further limit RNAi to the title field, after RNAi, I would type bracket, title, and you see that it is now narrowed to 10 papers that have RNAi in the title. You can see RNAi bolded in the title field of each paper. Okay, so we can see how to limit to very specific fields. We can also see how to use the AND operator. Be sure to use all capital letters for all Boolean operators so that it doesn't look for the word AND as a search term. There is another way to search PubMed by subject. That involves using the MESH database. MESH, which stands for Medical Subheadings, uses a controlled vocabulary. It allows for more consistent, complete searches. Some writers use different words to describe the same phenomenon. The controlled vocabulary is used to index each specific paper consistently. There is a very good video on YouTube provided by the National Library of Medicine, and it is available at this address. And you can view that video called Using Mesh to Build a Better PubMed Query for more information. Quickly though, if I want to search RNAi, instead of PubMed, I would now search the MESH database. And you see that the preferred term, again, this is a controlled vocabulary, is RNA, small interfering. If I want to search PubMed, I would simply add it to the search builder, and maybe even add and Mello bracket author, and then click Search PubMed. I actually got 25 papers, some different than the ones I got using the same search with just RNAi. That's because sometimes the term RNAi might not have been used in those particular papers, but those papers are about RNA interference. Therefore, I get those additional papers using the controlled vocabulary. You might also notice that on the left menu are a series of filters. Those filters include article type, so if I want only review papers, I would click Review, and that would narrow it down further. A popular one is text availability. Most papers, when you open the record, it only shows the abstract, but most papers have a link to full text, and sometimes the full text comes at a cost from the publisher. Sometimes it's free, and very often you would want free full text. Why not? If I click the Free Full Text Available filter, then I narrow down to 16 papers. Now, when I click that first paper, I know that there is a link that reads Cell Press, and it says Free in PMC in the upper right corner. When I click that link, I am going to be able to download the entire paper for no cost because I chose that filter of free full text. That's very useful if you are not at an institution that subscribes to a bunch of specific journals that would allow you to have access to them. The truncated search can be very useful if there are variations of a word. For instance, if you're unsure of spelling or if there is a singular plural issue, an asterisk can represent the rest of the word. The asterisk can only come at the end of the word and not at the beginning or the middle. Immunoglow asterisk will hit both the singular immunoglobulin and the plural immunoglobulins. You don't need to search twice. It's also very useful for an author name if you don't know the spelling. However, there is a limitation. It only shows the first 600 alphabetic variants. For instance, if you are using the name McDonald, and you're not quite sure if it's McDonald or McDonald's son, then it is better to type MCDON star than just MCD star. Put in as much of the spelling as possible. An example of that particular type of search is to type Immunoglow star and Smith bracket AU. I now get papers on immunoglobulin and immunoglobulins. The last topic to cover involved the date fields, both EDAT for entry date, meaning the date that that paper was entered into PubMed, or PDAT, publication date, meaning the date on the journal itself, for instance, the January 9th issue of Science. Ways to limit to specific dates include year. 2014 will limit you to the entire year 2014, or 1998 for the entire year 1998. Year/month. So 2014/03 will give you all of March 2014. Finally, the specific date. 2014/03/17 gives you March 17th, 2014. It's most commonly used in a range of dates. 1998/02 : the colon indicates a range. 2013/09/27. That translates to February 1st. Since it's at the beginning of the range, you get the whole month of February. February 1st, 1998 through September 27th, 2013. An alternative type of date search is last 19 days in quotes, bracket EDAT. Of course, the number can vary. So, for example, if I had heard about a paper that was submitted to Nature within the last 10 days, I would type last 10 days, in quotes, bracket EDAT. I know that it was submitted within the last 10 days, not with a publication date within the last 10 days. and Nature bracket journal. That limits the search to the journal Nature. Well, here is a good lesson. I still have a filter activated. The filter is free full text available, and Nature doesn't have free full text, so there are no results. I have to clear that filter because those filters persist. So if you use those filters, be very, very careful. They stay on in subsequent searches unless you turn them off. Now, if I want to limit to a particular date range, let's say we know that it was toward the end of 2011 or the beginning of 2012, somewhere around that time frame. So let's go for all of November 11 to, well, let's get all of February 2012. So let's type 2011 / 11 colon 2012 / 02 bracket e-date. Let's say it's a science paper, so capital A-N-D, science, bracket, journal. And it was something about DNA, so I'll put in DNA. I found 37 papers that were in the journal Science that were about DNA and sorta toward the end of 2011 or beginning of 2012. Maybe I could now sort through and find the paper I was thinking about. This concludes our exercise on PubMed, and I think the best way to really learn it is to go to PubMed and play around a little bit with some of these field tags and Boolean operators and see what you come up with. Good luck. Hi, I'm Bob Lessig. Welcome to our session today. And what we're going to look at now is a DNA record in the NCBI sequence database. In this case, we are looking at the Homo sapiens breast cancer 1 early onset. That's the BRCA1 gene that's associated with breast cancer risk. It's this transcript variant 1. There are several different transcript variants of this particular gene. But we'll look essentially at the DNA sequence of the BRCA gene and the protein that's associated with it. So this is the actual record. You see Homo sapiens breast cancer 1 is the title. And if we look at the top line, we see that there's an accession number, NM007294. It's actually a secondary key that identifies this particular sequence. It's 7,224 base pairs in length. It is an mRNA sequence, meaning that this is a spliced gene. The introns have been removed. It's a linear sequence. Really, only bacterial sequences would not be linear. The PRI means this is a primate sequence, so it's in the primate division of GenBank. And it was most recently modified on April 17, 2013. It doesn't necessarily mean when it was submitted. It means that was the last modification of this particular record. On the left, you see locus definition. These are all fields. And in the definition field, you see what's in the title. This is Homo sapiens breast cancer 1 early onset transcript variant 1 mRNA. You see its accession number with a version number. That means this is version 3 of the sequence. So basically, every time the sequence is changed by the author, you will keep the accession number, but you'll get a new version number. So it looks like this sequence was changed twice. The GI or GenBank identifier is a primary key. It's a unique identifier to represent this particular record. If a sequence is changed, the accession number gets a new version, but the GI number changes. And what we're going to notice is a pattern there that suggests that this is a primary key, accession is a secondary key. The keywords field is usually blank or some identifier goes in there. It's an obsolete field, but it's a required field in the database, so it's can't take it away. Its source is the organism it comes from, Homo sapiens. In this case, here is the taxonomic information on Homo sapiens going all the way back to its eukaryotic domain through its phylum and order and class, etc. You see a few published references relating to this particular sequence. And with the BRCA1 gene, you're going to find a lot. So those references are constantly updated. And you see that there are many references for this particular gene. Often one of the references is the direct submission of the sequence itself. And under the comment field, you see that this is curated by NCBI. You see that on May 19th, 2009, this sequence replaced version 632.52871. So you see it's a nuclear phosphoprotein. You get some comments in the comment field about what you're looking at in this particular DNA sequence. Then you see the features table. Features table tells you the source. This is Homo sapiens. The molecule type is mRNA as opposed to genomic DNA, which would be the unspliced version. It's on chromosome 17. We'll see a few synonyms for the genes. Sometimes it's called BRCC1, some historical names that may no longer be used, or in some cases, for some genes, there are researchers that call it one thing while another group of researchers call it another thing. So it's useful to have those synonyms there. You see some links to some other databases, and I'll show you some further links shortly. It lists a few of the exons. Exon 1 goes from position 1 to 213. Exon 2 goes from 214 to 312. Most importantly is the CDS region. And CDS is a quite unusual abbreviation. It stands for the coding region of a protein, beginning with the start codon and including the stop codons. And stop codon, even though it doesn't code for an amino acid, is considered part of the coding region of the protein by definition. And you may wonder how coding region becomes CDS. Well, coding sequence. Like I said, it's strange. So CDS stands for coding sequence, and that means that from position 233 in this DNA sequence to position 5824 represents the coding region. Now, the sequence itself is below, and I'm going to click the link that says CDS, and when you click it, it takes you down to the sequence. So here is the DNA sequence itself. You see a series of numbers that suggest this is nucleotide 1, this is nucleotide 61. You see they're laid out in groups of 10. And when I click CDS, it highlighted the coding region of this particular mRNA. So what we're essentially looking at is the start of this mRNA is what's called the 5' untranslated region. It is not translated into protein sequence. The highlighted region represents the CDS, or coding sequence, beginning with the start codon, ending with the stop codon. And then after the coding sequence, you'll notice that stop codon is the last 3 nucleotides is TGA, which would be UGA in RNA. And after that comes the 3' untranslated sequence, which is the region of the mRNA after the stop codon that doesn't code for protein. Now, you may notice that there are T's here not used. This is an mRNA sequence, and by convention, U's for uracil are never used in nucleotide sequence databases. So when you see an mRNA sequence, what you're really seeing is the cDNA sequence. The cDNA sequence has the T instead of the U. So note that if this were really mRNA, all the T's would be replaced by U's. The UGA is a stop codon. Going back up to CDS, you see that region, and you also see there's a link to the protein that if I click that, that would take me to the protein sequence for this particular gene, which also was highlighted when I clicked CDS over on the right side of the screen here. And you see the translated protein sequence just in one-letter code for all of the amino acids. And you see some locations of other features of that particular gene. In this case, there are an awful lot of them, more so than the usual record. Now, going down to the sequence, this is called the GenBank format. You see where there are numbers. So if I wanted to find nucleotide 134, I would see, well, this is number 121. I see that from the left. There are groups of 10. This is nucleotide 131. So 134 would be this particular G. It's very easy to find very, very particular positions. However, if I were to copy and paste this sequence, it would have numbers in it. It would be very difficult to do analysis on DNA sequence with those numbers added. So for running algorithms and programs, the most useful format sequence is called FASTA. And if I go to the top of the record and click FASTA, what you will see is a description at the top, and the caret tells me that that's a descriptive line. And below that descriptive line is the actual DNA sequence itself, and that's the G's, A's, T's, and C's that you see here. You see it goes down a long way. If I wanted to get that in plain text, I could do that simply by clicking FASTA text. And now I could select all and run any type of program I would want with that particular DNA sequence. I also want to show you the revision history under display settings. If I click revision history, it will tell me the entire history of this record at NCBI. And if you remember, there were 3 versions of this sequence. So I'm going to scroll way down to the bottom where it will tell me that accession number NM007294, and I should mention that there is an underscore. You can't see it here. The accession number does include the underscore. There's no space in an accession number. This is NM underscore 007294. That's important for search purposes. It was first seen at NCBI on December 9th, 1999. And then the sequence is changed. And usually when the sequence is changed, it usually means they finish sequencing the entire mRNA as opposed to just the coding region. On, it looks like May 11th, 2005. And then the new version was assigned and a new GI number. Gives you an idea of how many sequences are in this database. And it looks like version 2 persisted until May 19th, 2009. And if I wanted to compare what was different in version 3 and version 2, well, I can highlight version 3 and version 2. I'm going to click the left button on the 3 and the right button on the 2. And what will that allow me to do is make the comparison. And I can basically see what changed. It looks like I went from a 7,191 base pair sequence to a 7,224 base pair sequence. And you can see that the pink represents version 3, the gray represents version 2. Okay, now quickly I'll go back to the GenBank version. The default version, I should say, is called GenBank. Now I'm back to the GenBank record. And I want to point out that on the right menu, and they used to be at the top, but unfortunately they've moved them. It's a little hard to find. You see related information. And this will take me to other records at NCBI that are associated with this particular nucleotide sequence. So for instance, if I want the protein that is coded for by this particular nucleotide sequence, I could click protein. And now I have what looks very similar, breast cancer 1 susceptibility, protein isoform 1, except this is no longer the DNA sequence, this is now the protein sequence. I can see that by scrolling down to the sequence and note that there are a lot more than A's, T's, G's, and C's here. There are only 4 nucleotides, but there are 20 amino acids, so I'm using a lot more letters now. Protein sequence is 1,863 amino acids long. But otherwise, the record is set up pretty much in the same way. You see the references, you see the comments, and you see the features table. Now, I'm going to show you a particular region. Now, the BRCA1 protein has what's called a ring finger. Sometimes it's called a ring domain. In this case, they call it a ring finger domain. Sounds like a very interesting domain in proteins. You may wonder where they got the name ring finger. Well, it tells you right here. Ring stands for really interesting new gene, if you can believe that. So here's the ring domain. And it was originally called ring because of really interesting new gene, but the name stuck. And it's a very particular motif that's found in several different proteins that this one happens to have. It's from region 23 to 68. Now, just like I showed you the CDS in DNA, I could click region, and it will highlight the amino acids that are in that ring domain. And give me a little menu here to describe a little more about what that is. And just like the DNA sequence,
I've got a series of links on the right menu. I can get the sequence in FASTA format by clicking FASTA. Here it is, except this is protein and not DNA. And I can get the revision history simply by clicking the display setting for revision history. In this case, there was only one version of the protein, so where the DNA sequence changed, the protein sequence apparently did not. That tells me that the changes were made in the untranslated region, not in the coding region. Or if there were changes made in the coding region, they didn't affect the protein sequence. This sequence was also first seen at NCBI on December 9th, 1999. It's very common that the protein and DNA sequence are submitted together. And all of these changes have been to parts of the record that are not the sequence itself, meaning that perhaps a reference was added or some minor information was added. We see another whole series of links, and there are a lot of them there, and hopefully as you explore some of NCBI, you'll be able to use some of these links in the future. But that concludes our tour of a sequence record for now. For more information, please contact us in the Center for Biotechnology Information. Thank you.